Our next speaker is Andrew Steinsapir from Apertura. He's going to talk about the transferrin CapX gene therapy development.
Thank you so much. It really is a privilege to get to be here, and following up that talk is hard, so thank you for doing the hard work of walking through AAV. My name is Andrew Steinsapir, I was said earlier. I'm the acting chief technology officer for Apertura. I also work at Deerfield Management, which I'm contractually obligated to say. We work in healthcare development. The story that I'm going to walk you through here is one of what the company is doing right now and what our goals are overall. The most important goal that I want everyone to walk out of here knowing is that we're happy to collaborate and make sure that our technologies are accessible. We like the idea of having a big tent and actually having people use these technologies and not hide these things, hide our light under a bushel. For these efforts, I'll be walking through what we're doing right now with our lead and flagship capsid, which we named TfR1 CapX. I'll show you some of that data. I'll talk about ongoing work as well as why we like this mechanism. I want to be clear, I like all of AAV, I like all of cell and gene therapy, but I would love for us to not need it. I would much rather be in a world where we have small molecules that meet the needs of patients today, or ASOs doing the same. So I'm happy to be out of a job one day. With that, what we're doing at Apertura is actually starting in a little bit of a different approach. Rather than taking a random walk towards an optima, we're developing our capsids the same way that you would develop an antibody, and that's actually with your target in mind. Thinking about that mechanism that drives you to where you're going and why you are going there to help think about this delivery story. Our scientific founder is Ben Deverman, who developed PHPB and EB, which were mentioned earlier, and if you want a nerdy conversation about that, I'm happy afterwards. But what we do is we think about that as our starting point, select a receptor, develop a capsid for that. Our lead capsid uses the human transferrin receptor because it is known and validated for bringing things across the blood-brain barrier. Our goal was always to use IV-delivered products rather than brain surgery. We have a number of efforts moving forward right now that I can walk you through. And then overall, our goal is to make sure that our products are accessible. I don't have a slide on this, but just as a reminder for anyone that's an academic in the room and for others, we have an open MTA process that's about 3 clicks on our website, and we will make sure that you have access to the capsid for research purposes. CapX as an overall is doing something that I think a lot of people have seen pictures of before. On the left-hand side, we have AAV9 with an IV dose of 2E12 viral genomes per kilogram. And on the right-hand side, we have the same thing, but with CapX, and these are both in mice that have the human transferrin receptor expressed, knocked in. Our scientific founder put out a publication in 2023, 2024, talking about our first generation capsid in Science. It was the first time ever anyone had actually been able to target the human transferrin receptor using AAV, which had always been a target. The thing that we're using now, TfR1 CapX, is the improved version of that, and truly, I think, as good as we need to be for moving these ahead, because we're always going to have that tension between improvement versus getting things to people that need it now. We've been able to meet these metrics that are here, but rather than walk you through that, I can actually show you on the other slides. What we think this means is that we have an opportunity to help unlock neuro and neurometabolic disorders and actually bring things to bear. We've seen the capsid be used across the board with payloads in the gene replacement or supplementation space, RNA transcript regulation, epigenetic editing, split inteins for things that are too big, gene editing. If it fits in an AAV, or even if it fits across multiple AAVs, you can put it in this as well, and we're happy to help with that. What that means in my mind is we have an opportunity to try to build a step and repeat platform to whatever degree we can in getting things to the brain. The biology of each of these diseases is going to be unique for each of these diseases, which is why we need people learning and understanding these things. We want to make sure that we actually have the tools and the know-how and share that data and the benchmarks that we've already set so folks don't have to spend their time reinventing the wheel. Now with that, we have our own programs. We're not just out here giving things out for licensing sake. We're also developing therapeutic products internally because if our stuff's that good, we should be using it. But we're also making sure that we work with groups. A few of those are going to be moving into the clinic in the next 12 months. We're really excited for them. We work closely with them. Those are the group Finding Hope for Frizzle that you see there at the top. They're moving forward with a rare DEE. Aviato Bio, that's going in with a knockdown approach for MAPT. And then our collaboration with Broad Institute with Sonia Valab looking at prion disease. We have other groups that have already disclosed that they're working with us, but we can't show that they're pipeline yet, and others that aren't disclosed, but happy to talk about what this means. Our general approach in licensing is to make sure that we're doing things non-exclusively because we don't want to be picking winners. We want to enable people to move things forward. The mechanism that we selected and went after human transferrin receptor for the neuroscientists in the room, which I realize this is probably an indexing on neuroscientists, and I am not one. So all of you know very well that this is the right target for bringing protein across the brain because that's actually what it does normally. And this well-validated mechanism that we are standing on the shoulders of over 40 years of research and work for is one that's also already being used in clinical trials and approved products in the biologics space. We have the opportunity to use a receptor that is not only well conserved in how it is expressed and where it is expressed, but one that's consistent across mammals and age. For our efforts and our work, we like that this receptor is always engaged and is always cycling and is always moving protein across so that it's actually in a current moving across that it can bring either an antibody or our products forward. With that and thinking about what the difference is between doing that with, say, an AAV versus antibodies, what I have on the screen here are 3 of those products that are either approved or in trials, the first 2 being the approved products, which are both in Hunter syndrome. JCR's product is an IgG with a fusion. Denali's is just that FC portion with a fusion. Both of those are approved for pediatric use. And then we have Roche's trontinemab, which is an IgG with an extra transferrin targeting arm. I'm a nerd, if that doesn't show. I'm an engineer, which means I think about length scales a lot. AAV is big, but it's actually not that big in comparison to other things. So antibodies themselves are pretty floppy and they take up a lot of room. And as a result, we're in a similar size regime between antibodies and AAV for the space that they take up. And the thing that says to me is that we're not actually asking this receptor to do something or pick up something that we haven't shown it can pick up before or move across before. The dose levels that we're looking at from a multiplicities of engagement for this receptor are orders of magnitude below what is already being demonstrated as safe in pediatric and geriatric populations for antibodies and under repeat administration versus a one-time. We have not put this into a human yet. That will be happening in the future. It's something that we take very, very, very seriously. There's no one experiment that can guarantee safety. We all know that. What this does allow us to do, though, is think about the precedents that do exist with these products and the information that we have. Trontinemab being one that I follow very closely because those patients are receiving serial MRIs throughout the course of their treatment. The way that our capsid works is we actually bind to the same area or similar area as those antibodies. We don't interfere with the natural ligand that transferrin receptor uses, so we're not displacing something or competing with it. We're actually just piggybacking. It's almost like we're carpooling. What we know for our capsid is how it interacts down to the amino acid. We've always been open about this. We know that that amino acid is unique to humans. There is no laboratory species or non-laboratory species that has this. As a result, we rely on human transferrin receptor knock-in mice to do definitive studies for distribution and toxicology purposes. We have also executed studies in large animals to look at the impact of the capsid in them, but we are not able to see pharmacology for distribution to the brain in them because they lack the correct receptor. What we have been doing and continue to do has been showing us that we can achieve full brain transduction at levels of about 2 E12 viral genomes per kilogram. That's a weight-based dosing, getting about 80% or more of neurons in the brain. So this is about 50 times lower than the dose levels used for the approved AAV9 or RH74 products that are out in the market. That significantly reduced dose means that we're exposing a lot less product to a patient, and in my view, the less we use, the better, and I think everyone has said that so far. With this, what you're seeing on the screen is the initial AAV9 that this capsid is based off of, the first generation capsid that was published in Science, and then TFR1 CapX. We have to also keep dropping the dose levels because otherwise we blow out the images when we're doing these. And what you can see here is some of those comparisons and what tissues we're moving to. I'm someone that's worked on a lot of neurometabolic disorders. For me, it's actually been important that we're able to reach liver because there are diseases where we need to be both in the liver and in the brain. And as a protein engineer and AV capsid engineer, it's much easier to take away an ability than to bring it back. So we made sure that when we were developing this, we had that drug development idea in mind that there are going to be a lot of people using this, not just us for this one thing. Our work is consistently comparing itself to using mouse surrogate capsids like we saw earlier. The message that I would take or use as a takeaway here is if you can achieve delivery in a system with PHP-EB, we can beat that out by about an order of magnitude, which helps us when we're thinking about feasibility and therapeutic development of these products, of going from being excited about outcomes in a study to thinking about translation. Our work that we're showing here is just a little snippet of this, but overall biodistribution that we see as a consistent unlocking of the CNS, as we see a reduction in exposure to tissues that are non-CNS. Human transferrin receptor, or transferrin receptor in general, is expressed on muscle as well as on the brain vasculature. What I believe that we're actually seeing, and this has been replicated by others attempting to follow in our footsteps with transferrin receptor and AAV, is that on the length scales and with AAV, it is actually only the brain that has a mass transport barrier that's holding back transduction. It's not getting an enhancement in muscle because that's not the limitation that we have. We also know from unpublished data that will be coming out shortly, I hope, if someone finally gets back to review comments, of what the kinetics are of this capsid and it coming out of the bloodstream. With this, we have made these things to make sure that they're boring and mundane, which means manufacturing is also boring and mundane and something that you can do based on what's on the back of the box. It uses all standard approaches, which means you don't need bespoke methodologies to move them forward. We've been able to scale this already and we have clinical manufacturing lots and scales that have completed successfully at multiple parties. So it's not just working in our hands or in Ben's lab, but now dozens of people's hands, and we're excited to see that. The team that I lead can also help come in and help move those things forward, and it's something that we're excited to see the field adopting. Just a summary of all of these, of what we're seeing as these efforts are moving forward, and then I want to get to the slide that I actually think is both more meaningful and exciting. We work a lot with a number of groups. One of the ones I am allowed to talk about is the work that we do with Sonia's lab, focusing on prion disease. For folk that are familiar or not familiar with prion disease, it's a neurodegenerative disorder due to a misfolding of protein. Prion as a protein is only made in neurons, but we do have mRNA that is expressed in every single cell in the brain because brains are weird. So what you're seeing here is in situ hybridization, or ISH, for the mouse prion mRNA. And on the left-hand side, you're seeing that basically everywhere in the brain has it. And what you're seeing on the right-hand side is what those brains look like after a single dose with this capsid and their knockdown strategy, which is an epigenetic editor. I live in New York right now, and this reminds me of what a blackout looks like when the lights in the city go off. For prion disease, we actually have a lot of information because of the work that Sonia has done and led in the field of how much reduction we would need to have a positive benefit in these patients, and that's about 30 to 50%. We're already seeing much more than that at these doses and below, so we're excited to see this move forward and collaborate with them. The pipeline, which I already showed you, is something that is expanding moving forward. We're excited to see more folks be able to leverage this and move these ahead. And then the thing that when it first happened, I called my parents to tell them, I'm excited that our first partnerships were actually with rare disease foundations, not with companies. So we partnered with the Rett Syndrome Research Trust first as a public announcement, and then we've brought on a number of groups since then, including the ones that you see there on the screen. Our effort is to make sure that this isn't just a large pharma company doing this licensing or a biotech, but also patient foundations, academic labs, N of 1 families, because God forbid we help someone move these forward. The tools that we have for the scientists in the room, because I heard a lot of people talking about iPSCs. Most iPSCs will express human transferrin receptor natively. The capsid works a lot better in vitro as a result than AAV9 does, or PHP-EB, because that's mouse and for a different receptor, which can make your life a lot easier when screening payloads. That's not normally a pitch I would give when trying to get someone to use this, but it means that if you are evaluating or working with iPSCs, even if it's not to do development in AAV, happy to make sure that you have access to this, because it's a lot easier to use, and we're getting wonderful looking transductions at MOIs of like a double-digit number or triple-digit number, not a 5 or 6-digit number. With that, I have a standard slide about collaboration, and I'm happy to take any questions should there be any. Thank you.
So, wonderful talk. So the question is, there are many therapeutics that are with antibodies, and to reduce the protein, and you showed us now that you clean completely the with a prion disease. So the question is, are you going towards this direction, like in cancer and also in Alzheimer's disease, all these developments?
Thank you for the question. So many people are already using human transferrin receptor to bring large biomolecules across the blood-brain barrier, and I think that they're using things that do make sense to get there and using effectively protein replacement therapy. If one was to go after that with AAV and do protein replacement therapy, I think that's also viable. On a personal side, though, I think if we have an antibody that's working and is effective, we don't need to be going into gene therapy necessarily. For our efforts and the things that I'm overseeing right now, I'm seeing a lot of NDD, DEE, and then lysosomal storage disorders. But frankly, this is just one of the many areas that we're working on, and we're happy to collaborate across the board. We don't really have limitations in how we make sure people have access. I will note we do work in TSC. Even though we're doing work in TSC, we're actively making sure that others working in TSC can have access to this capsid and move things forward, because we want to know that moves forward for that community. Thank you for your question.
Yeah, thanks.
Hi, Bev.
I have a question. I want to challenge you on your
Please.
comparison of size.
Yeah.
Where I'd like to know how you did the math on 150 kilodalton antibody taking up the same space.
It
as
is
a 4 megadalton.
Absolutely, it's hydrodynamic radius. So because of the way that antibodies actually are moving and flexing consistently, the space that they take up when we do hydrodynamic radius, it's on their movement itself, is much larger than you would guess from a globular protein.
Correct, but I'm not talking about an antibody that's moving. I'm talking about an antibody that's bound to a receptor and then internalized, where the physical space is quite a bit different. And then I'd like to, just to compare and contrast there, where really it's probably more than a tenth of the space within the cell that an antibody is taking up once it's bound and internalized into the endosome. And how do you consider some of the issues with the fact that AAVs undergo, as a natural part of their process, endosomal escape, and the fact that you will be transducing a large part of the endothelium? I think we saw that pretty effectively in Ben's first science paper.
So first science paper is the first generation of the capsid. Thank you for that. On the endosomal escape portion of that, that also does happen for the antibodies themselves. When we're engineering these to use the human transferrin receptor, we do, I'm going to get real nerdy here, we do pH modulation so that they can come off and on so that they don't get fully degraded, because we need them to pop off. In actuality, when they're bound to the receptor, they're still moving. They're not bound and locked. We know that because we have data with both ASOs that are conjugated and others, so they're taking up much more space than just their bit. It's always going to be dynamic. The size of those, I don't want to use the word vacuole because that is the wrong word, but those, well, I guess they're endosomes at that point. Those endosomes is large enough, regardless of whether or not it's an antibody or an AAV, that they're going to be taking up sufficient space. Without that being limited, your last one about hitting vascular endothelial cells and that transduction, we have data on that, and across the board, we see a considerably lower transduction level in vascular endothelial cells when you use a receptor that is actually meant for transcytosis than when using, say, a GPI-anchored protein or a protein that isn't meant for moving that forward. I think there's also a question here on some of the engineering aspects that can improve those outcomes, which I think Ben will be describing in more detail in the follow-up paper, but I think he's been doing a lot of prion work instead. But happy to talk. Thank you so much for the question.
Hi, Andrew. It's exciting. It's also, frightening, I think, almost.
It's terrifying.
And maybe this is a tough one to answer, but I just want to hear your thoughts. Moving into first-in-human trials without doing biodistribution and safety in a large animal model. How do you inform that dosing in those initial human trials? Weighing both prospect for benefit and minimizing risk. Quantitatively, how do you all think about that? How scaling up? You're talking about these length scales, of course, when the construct hasn't been tested in something larger than a mouse, perhaps.
Great questions. So one thing to hit and clarify: we have done studies in large animals. We're not required to execute studies in large animals iteratively. That's more of a platform-based outcome rather than an every product-based outcome, because we don't have delivery opportunities, or because there isn't on target. For thinking about scaling dose, just taking that as a separate bit, since the length scale that we're always crossing regardless of mammal is about 20 micron, that we're going across. We're actually taking a page out of the book of folks that are working on this in the antibody space as well, where that mass versus weight ratio can remain constant, and we see that exposure for both PK and distribution in animals and in humans. So JCR's product is approved in Japan. On the PMDA's website, Japanese FDA's website, they have 2 copies of their approval. There's one in English because I do not read Japanese very well. And in that they walk through their ADME and PK. And it's actually only in their mice that have the human transferrin receptor knocked in that they have representative PK of what they saw in people that helped inform that dosing. Now, with that, does doing that mean that you absolutely know what's going to happen? Not at all. Every single model we use is just that. It's a model. When thinking about these moving forward for different diseases and different payloads, I think there are different risk factors inherently in how we look at these in the systems. I think some of the presentations earlier actually walked through that and how to think of it from overexpression risk as its own, and as well as things like immunotox or hepatotox, and these other capsid-based outcomes versus payload-based outcomes. Both of those have to be evaluated. I would say that there is a important distinction and difference between what is sufficient information from a regulatory perspective to move forward versus what is sufficient information as a sponsor to feel comfortable, or as a community, or as a clinician to feel comfortable on that. And that's something that we spend a lot of time working directly on. You'll notice those 3 programs that are moving forward are not ours. We're working closely with those groups as they move ahead, and each one does take a different tactic to that. I would say overall, though, we do get the consistent set of feedback here on making sure that we're looking at each of those points that you brought up. I don't know if that satisfies your question, though.
I have more, but I'll follow up.
No,
later,
yeah, I'm
after
happy
the
to. I'm
session.
here through tomorrow. Thank you.
Thanks.
Thank you.